All right. We have now talked about every possible taxonomic permutation of protists. Wow, that was hard to say. Permutation of protists. We've talked about what these things look like. We've talked about the different classes. I want to talk about the consistencies between them, how they acquire nutrients, how they defend themselves, and how they reproduce. There are 4 possible nutritional adaptations in protists: phagotrophy, osmotrophy, photoautotrophy, and the aptly named mixotrophy. We'll go through each. Phagotrophs. They take in food through phagocytosis. That is, you're going to have food brought in across the membrane using vesicles. So food particles come in, there's a change in the membrane, that membrane closes a vesicle around the food particles, and a lysosome will then bind to the vesicle to digest it. That's phagotrophy. Any questions on it? Osmotrophy is where you take in food directly across the membrane. You remember osmosis? Remember diffusion? Remember how diffusion, you can move from one side of a membrane directly to the other side through channels? That's what's going on with osmotrophy. You're moving materials from one side of the membrane to the other. You are an osmotroph. You eat food. That food passes along the cells of your small intestine. And as they're going by, the cells are directly absorbing the nutrients. They're not sucking them into vesicles like you would as a phagotroph. So you're moving food directly across the membrane. Photoautotrophs, we've talked a lot about these. Photoautotrophs make their own food particles from light. They're going to create organic molecules using inorganic molecules. And then you have the mixotroph. Mixotrophs are a combination of either phagotrophy, osmotrophy, or photoautotrophy for their nutrient gathering mechanisms. So those are your 4 big concepts: phagotrophy, photoautotrophy, osmotrophy, and mixotrophy. You can have lots of different types of defensive adaptations. You can defend yourself. Well, protists defend themselves through either mucilage, extrasomes, bioluminescence, or toxins. First off, you got mucilage, mucilaginous protection. There is a shield around the cell that acts almost like an extra membrane layer. It protects the cell from being dissolved or from being broken apart. Further, mucilage can hold 2 cells together for mutual protection, like glycocalyx, just saying. Extrasomes. Extrasomes are used by cells to release internal components of the cell into their immediate environment. Some microorganisms might use almost neurotoxins to do this. In fact, the jellyfish, the nematocyst of the jellyfish, uses extrasomes to take that internal structure and make it external. Literally, it spears you. It sticks you. And then as it spears you, it releases a toxin, which of course leads us to our final category. It's not final, sorry. We're going to talk about toxins in just a minute, but some organisms are bioluminescent. They light up. They do this to startle predators. They might do this, some of them actually do it in order to, once they're eaten by another fish, they light that fish up so that another organism eats that fish. There are algae out there that when they're disturbed, they do a flash in order to warn other organisms around it. If you're at the beach and it's a dark night, We're looking at, no lights on shore, and there's no moon. You can see bioluminescence in the water. The water looks like it's lit up. The moon jellies, for instance, right? There's a lot of times an association between protists, microorganisms, with bioluminescence and the larger organism. Most of those organisms at the bottom of the sea that you see lighting up are not lighting up because of them. They're lighting up because there's microorganisms living inside of them. And toxins. These are secondary metabolites that disrupt the biological systems of other organisms. So there we go. Big defensive mechanisms are extrasomes, sort of these internal organelles that can release their contents to the outside of the cell. Toxins, secondary metabolites that inhibit growth or stop other organisms from surviving. Mucilaginous shields, or bioluminescence. Protists are wildly prolific. That is, they can reproduce an awful lot, and they do it in several different ways. They can do it asexually or sexually. We've talked about prokaryote cell division, and it was entirely asexual, all through binary fission. You make a copy of yourself. Protists are a bit more complex. They can, in fact, undergo binary fission to create identical daughter cells. That happens, but that doesn't lead to a lot of genetic variation. And if there's one thing protists are known for, is their genetic variation. Some protists, they're not going to reproduce through cysts, but they're daughter cells that, even though they're absolutely identical to the parent, they have what's called a thick peptidoglycan wall to protect them from harsh conditions. Where have we seen peptidoglycan before? With bacteria, when we were talking about gram-negative and gram-positive bacteria. It's that outer layer that surrounds bacteria, and it can also surround protists. It gives them some extra protection. Cysts can survive for a very long time without water, without food. It's like they go into a state of suspended animation. So you've got a daughter cell that's just like you, except can survive easier in harsh conditions. Protists might reproduce through what's known as zygotic sexual reproduction. They create two haploid cells, and those two haploids fuse to create gametes. When you make a haploid cell, what biological process are you using? It starts with an M. Meiosis. And meiosis leads to a lot of genetic variation. So you split yourself using that meiotic process, and then you get recombination. You get all the crossing over, so you have new variation introduced during zygotic reproduction. They fuse to make a zygote that then, in turn, can undergo mitosis. You might undergo a sporific process. This is also called alternation of generations, where you've got one, we're going to go into a lot of detail about alternation of generations in the next few weeks. I will tell you this will be one of the major essays on your exam when we get to it in depth. This is just your first introduction to it. The basic idea is you start off with a zygote, a 2N individual. That 2N individual reproduces via mitosis to create identical daughter cells. That creates a sporophyte stage. The sporophyte undergoes meiosis to make genetically distinct daughter cells. So far, so good? The genetically distinct daughter cells are called spores. The spores themselves, the haploid spores, undergo mitosis to make many, 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 many haploid spores that are all identical to each other, which are then released as gametes to form the zygote. So it's going to be alternating between a diploid stage and a haploid stage. That's why it's called alternation of generations. You're alternating between diploid and haploid. We will talk about that in greater depth when we talk about plants. Gametic reproduction is really interesting. In this case, you have a cell in a group of cells divides into haploid cells, which then fuse with the cells next to them. Literally, you got cell and a cell and a cell, right? N, 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 N. They're all 2N. They all have, they're diploid. Make sense so far? What they do is they create gametes, and the gametes fuse with their immediate neighbor. It's like a horrible round from square dancing or something. But now you end up with new combinations because crossing over has been able to occur, because independent assortments happened. So that's gametic reproduction. So we've got sporific, zygotic, gametic reproduction, as well as binary fission. Ciliates are interesting creatures. Ciliates are like paramecia. They have these structures in them called macronuclei. They're not the nucleus, but they are groups of DNA, like little balls of DNA that they can pass back and forth. They come together, they fuse to each other. So instead of splitting, they literally come together, become one organism for a bit, pass those little DNA balls back and forth, like jugglers, and then split apart. It's sort of like if you were to mate with your partner, you've got blue eyes and they have brown eyes. At the end of the engagement, you'd walk away with their eye color and they'd walk away with yours. Just sharing that genetic material. Not creating an offspring, just sharing. Kind of interesting.